Science Positive, a podcast dedicated to sharing the stories of biologists and scientific investigations at the Department of Biology at the University of Washington. My name is Emma Guerrini-Romano, and I will be your host for today's episode. Joining me today is the wonderful Glorianna Gutierrez, a second-year graduate student studying circadian rhythms. Glorianna, welcome.
Hello.
Can you please tell us a little bit about yourself, where you're from, what brings you here?
So my name is Glorianna Gutierrez, and I am originally from Venezuela in South America. I moved to the U.S. when I was 16 years old, and I have been in Seattle since then. I did my undergrad at University of Washington. I got my bachelor's in neuroscience. And then now I'm doing a PhD in biology, studying circadian rhythms, specifically circadian rhythms in the immune systems on cell motility.
Awesome. So you went straight from your undergraduate degree to your PhD.
Yes, I did not take any break.
Wow. So is that something that's pretty normal, or do you feel like you...
I think that is more normal to take a gap year between your undergrad and your PhD. But people do go straight into their PhD. I think for me, it's like I knew I wanted to do a PhD. I was very clear on what I wanted to study. And I was lucky enough to get a lot of research experience during my undergrad. So I just felt like it was a better use of my time to jump straight into a PhD, especially because it takes quite a long time to get a
awesome.
PhD.
Yeah, totally.
So I just felt like it made more sense for me to jump straight into a PhD instead of taking a gap year.
So you are technically an alumni of the University of Washington.
Yeah.
Go Dogs. So you decided to stay at the University of Washington. Yeah. Are you working with the same PI that you worked with previously? Did you join a new lab? What does that kind of look like for you?
I was originally going to be staying in the same lab that I worked during my undergrad. I studied circadian rhythms. That's like the number one thing that I knew that I wanted to study. And there is not that many people who study circadian rhythms. I feel like it's not as common. So I was planning on staying in the same lab. But the first year of my PhD, it was quite an interesting year. I learned a lot about myself. I learned a lot about what I wanted to do. And I got to also work in different labs. So for those that don't know, during your first year of your PhD, you normally do these things called rotations, in which each quarter of the year you will be working at a different lab. And you are learning about different forms of mentorship and different skills and things like that. So during that year, at the end of the year, I just felt like what I was doing before wasn't necessarily what I wanted to continue doing for my PhD. And I had a very nice experience with this other PI, Julie Theriot, which studies cell motility. And I did a rotation with her over the summer, studying circadian rhythms in these cells called neutrophils, which are a type of white blood cells. So it's part of your immune system. And I just really liked it. It meant working with zebrafish. So it was a totally different model organism that when I was studying before, because during my undergrad, I worked with mice, different skills, a lot of microscopy, which I have found that I really, really like. So I just decided to stay in the lab because I just felt like it fit what I want to do long term a lot better.
Yeah, that makes total sense. And it's great that the university has a system set up to kind of protect grad students in that way, right? So we can do what we decide. I guess kind of on that related note is circadian rhythms is your bread and butter, I would say, which is great because I did not sleep well last night.
Neither did I, actually.
And I would love to know why. I'm sure that's a very complicated question with a lot of factors. But I guess the question I have for you is circadian rhythms, did you fall into it? Did you seek it out? Did you have any experiences with bad sleep as a child, per se, that traumatized you to study it?
I was very interested in circadian rhythms from a pretty young age. As a kid, I was always a very curious child, and I was very obsessed about why do we sleep. I hated taking naps. I did not want to take naps. And also, why do we dream? That was another thing that I was so interested on. And this was around the time in which YouTube became a thing. So I, as what probably a lot of us did, I will just go into YouTube and just look stuff and informations.
Yeah.
And I stumbled into this YouTube video who she did science communication in Spanish. And she had a couple videos about circadian rhythms, and I was what is this? And then I learned circadian rhythms is the study of why do we sleep? But also about other type of rhythms, what I love about circadian rhythms is the fact that like you can find them everywhere. Like basically everything in your body is being governed by circadian rhythms. Like when do you eat?
Yeah.
When do you sleep? Reproduction, like your hormones, like everything is governed by circadian rhythms, and that happens in humans, but also like it happens in plants and animals, and so it's just everywhere. And I found that very interesting because there are so many different things that you can study. So you really can get bored.
Yeah. Totally. So within circadian rhythms, what do you consider your specialty? What is your focus for your thesis?
Yeah. So right now I kind of pivot. I will say before, like I was studying like how our circadian rhythms like originated in the brain, and also like how it affects different diseases. So I worked with epilepsy during my undergrad. I had some experience working with Alzheimer's.
Wow.
Now I'm kind of switching a little bit and I'm working in the immune system, which is something that I have no experience with working before. So it's definitely new and I am excited to learn about it. But I think overall, I will say that I study how like circadian rhythms affect human health in some form.
Awesome. So I know that before you used to work with mice.
Yes.
And now you work with zebrafish, which if the people don't know, zebrafish are a very common model organism, very readily available for scientists. What are some of the differences between working with mice and zebrafish in the context of circadian rhythms? Obviously, you're asking different questions.
Yeah.
But what are the kind of methodologies that differed? Which do you prefer?
They're very different and very similar at the same time. They're both vertebrates, so that helps. I think a difference is that mice are nocturnal and zebrafish are diurnal. So zebrafish, I guess, have a circadian rhythm more similar in that sense to humans because humans are diurnal. A lot of human like health research specifically is done in mice because they're mammals, which makes sense. But there is a fact that mammals are nocturnal, so there are the fact that mice are nocturnal, so there will be some differences when it comes to comparing it to humans. So it's nice. I really like the fact that zebrafish are diurnal. I feel like that kind of makes that translation a little bit easy. What we call the molecular clock, which is basically like the genes that generate circadian rhythms, is something that is pretty conserved throughout different species. So it's relatively similar between zebrafish, mice, humans, which makes it nice and it's easy to study. There are some differences. So like zebrafish went through like genome duplication. So they have of the specific genes, sometimes they have multiple copies.
Whoa.
Which can sometimes I have been finding that it makes it kind of complicated when you're trying to do like when you're like, for example, trying to knock out and like mutate the circadian like genome, like it's just can be hard if you have like 4 or 5 copies of a gene. So those are some differences. I think that something that I really had to learn kind of like how to study is like I'm used to how to study circadian rhythms in mice, and you know, I was in a lab who has been doing this for like many years, and like the systems of like how do you record that activity and like how like the type of data you collect, like it was all very well set up. And then I moved to a lab in which they don't do circadian rhythms, and I kind of had to like figure it out, like, okay, how do I collect the same data on zebrafish from things of like, oh, like we need to control the LiDAR cycle, right? Like when is light and when is dark, and like, okay, how do I do that with the zebrafish? Like, you know, like those were all things that I had to like think about and figure out like how to do. So that was definitely interesting experience, but it was also a very fun experience to be able to like figure all like those things out.
You mentioned the word diurnal earlier, and you said that humans are diurnal. Can you expand upon that and tell us what that means?
So diurnal basically means it's for like animals, organisms who are active during the day. So basically means when we leave our active time, we eat, we do our activities during the day, and then we sleep during the night. So that will be a diurnal animal. A nocturnal animal is the opposite. Like a mouse is going to be sleeping during the day when it's light out, but it's going to be awake and going around and doing their business during the night. Yeah,
that makes total sense. That's a good way to put it. And so the opposite diurnal is nocturnal. Cool. So I guess a question for you that I have now about your research specifically is like throughout your PhD, you've shifted a little bit of your questions, your methodology, your approaches. So what kind of specific topics are you looking to answer with the neutrophils that are in the zebrafish? And how are you setting up your thesis for that? And what does it kind of look like for you after your thesis?
Yeah. So a lot of those things are still kind of trying to be figured out, but the main thing that I am looking at is so neutrophils, they move. That's like their motility is like a key component of neutrophils. They're always moving around trying to find things that they should be like getting rid of. They are like the first line of defense when like for example, there is a wound and they just like go fly to try to like protect it and just make sure that they like eat anything that they shouldn't be there. So their motility is like a key factor of neutrophils. And there has been studies that have shown in mice that there is more neutrophils present in the blood during the time in which the animal is active. So in the case of a mouse, that would be at night. So that seems to point to the fact that maybe circadian rhythms are governing like or immune system. So kind of the question we had was like, okay, well, there is more neutrophils present, but is motility also being affected? Because the lab that I'm working on right now, motility is like what they are the most interested on. And we also wanted to know if we saw similar differences on zebrafish. So the experiment that I conducted over the summer was basically to try to see if there was any circadian rhythmicity on neutrophil motility, but also just like neutrophil presence overall at different times of the day, comparing the time of the day in which we would expect the neutrophils to be active, which is while the fish is awake, so during the day, compared to when the neutrophils we will expect them to be not active, which would be at night. And I'm still finishing analyzing the data, but what my data seems to have suggested is that motility itself of the neutrophils, not just whatever their presence or not, but whatever they are moving, and they're more likely to be moving, appears to be under some form of circadian control.
Wow.
So the neutrophils early in the morning appear to be more motile compared to the ones that, in this case, it was like neutrophils that were in fish that were raised in constant light or constant darkness, so their circadian rhythm was being disrupted. The motility of the neutrophils was not as high compared to the ones that their circadian rhythm was actually like entrained and working correctly.
Wow, that's fascinating.
Yeah, it's very interesting. I still need to finalize analyzing the later time point, and that will really finalize giving us like if it's like I will expect to see also something similar in which in the later time point there is lower motility because we wouldn't expect the neutrophils to be active because the animal is not as likely to be exposed to any pathogens that the neutrophils have to fight, so that's why they wouldn't be active. So that's kind of what the basis of what I'm going to be studying. Some of the questions that I have is, well, one, is this, something that I can replicate with multiple zebrafish? But then also, like, what is the mechanism behind this? Like, what is generating this rhythmicity? And something interesting about zebrafish is that so in humans, our circadian rhythm is, we could say, entrained. We could think about entrained as like being regulated when it's exposed to light, right?
Yeah.
And our eyes, there are some cells in our eyes that are able to get that light information. And in zebrafish, it's interesting because every cell of their body is able to perceive light and is able to be entrained to
every
light.
single cell?
Yeah.
Even their like fin cells?
Yeah, like their skin cells are able to like collect. Yeah, so that is interesting because also makes things hard because you could, they are just very receptive to the light.
Sure.
So what I'm trying to study is whether the neutrophils themselves, like, is the circadian rhythms in the neutrophils, like, if you isolate it from the rest of the body, like if you, for example, like eliminate the circadian rhythm in the rest of the body, but only keep it in the neutrophils, is that enough to generate that motility rhythm that I observed, or does it require the circadian rhythm in the rest of the body to be able to do that? And that is a question that is actually trying to be answered also in mice, because for the longest time in circadian rhythms, we had discovered this area in our brain called the SCN. And that area, basically, we discovered that if you damage that area, the circadian rhythm of the animal is disrupted.
Okay.
So there was this idea that, okay, that's like the core circadian rhythm, and like that's like the center of circadian rhythm, and like that you need that to be able to like generate circadian rhythm. But then it was found that you have these things called peripheral clocks. So like for example, your liver, your heart, like other areas of your body have their own clocks. And in humans, what it happens is that these clocks need input from the suprachiasmatic nucleus to be able to have their circadian rhythm entrained, but they have basically their own circadian rhythm.
Okay.
But it is in humans and in mice, it is hard to like isolate them. So what I'm hoping to be able to study in zebrafish is that I'm hoping using the neutrophils, can I isolate them and isolate these peripheral clocks and like try to show whether these peripheral clocks are able to work on isolation, and are they able to entrain to the circadian rhythm on their own without any input from the SCN. So that's kind of like the big question that I have that will be applicable not just to zebrafish, but it could be applicable to other areas of circadian rhythms.
Wow, that's amazing. I mean, and you've done all of this in what, a year and a couple months?
Yeah.
That's rocking. So I guess a question that I have for you for follow-up is, so when we're looking at these circadian rhythms and you're doing studies in zebrafish, you're doing studies in mice, you said they're both vertebrates and humans are vertebrates. Check the spine.
Yeah.
So what kind of things from your research can we superimpose on, or can we translate to human health and to human sleep? Specifically, we're talking about neutrophils and immune systems, and so is there a correlation between circadian rhythm, sleep, immune systems, and human beings, and like what kind of things do we have to know for that?
Yeah. I mean, circadian rhythms in your sleep is like incredibly important. And I mean, there is, some research that has shown how like if you have like bad sleep and your circadian rhythm is disrupted, like it will impact your immune system. You're more likely to like get sick, right? So it is something that it could be relevant. Like all the research that we do, we always try to do it with this idea that like we can get something that will be helpful for humans. I think that right now, there is some evidence that can show like, like if you are sleeping terribly, like it's affecting every part of your body. Yeah. It can make your immune system be weaker, which will like mean that you're more likely to get sick. But why is that happening? That's like something that we don't understand, that my research could possibly give part of the answer of like, okay, well, if your immune system is also being regulated with the circadian rhythm and you're sleeping poorly and your circadian rhythm is disrupted, maybe you're now gonna like, instead of your neutrophils be active during the day, in which like they should be active, in which you're more likely to be exposed to pathogens, maybe now they're active at a time in which like you are not being exposed to pathogens. And the same thing you should be exposed to pathogens, the neutrophils are not active, so then you are now more likely to get sick.
Yeah.
So, that can be something. The other thing that I think is happening for circadian rhythms in humans is that now, due to like having artificial light, a lot of the times, like, what time do we go to bed, right?
Sure,
like I went to bed almost at midnight last night.
That's early for me.
So we're going to bed at times in which we didn't used to go to sleep before. So we are exposing our bodies to things, at times in which like our bodies aren't ready to be exposed to things. Like I think a big one is eating, for example. Like the time in which we eat, it's actually important. And eating late at night, there has been some research that has shown that there are negative effects of eating like late at night. So that's kind of the type of things that like circadian rhythm research in mice and in zebrafish are able to like kind of answer.
Wow, that's so cool. So I guess like in your day-to-day life outside of work, you leave the life sciences building at the University of Washington and you go home. Are you actively thinking, what am I doing that's affecting my circadian rhythm?
Yeah, I do. I mean, it doesn't mean like I am any better than everyone else. I mean, I still stay late at night with my phone.
Sure.
I still like go to bed at times that I probably shouldn't be going to bed.
Yeah.
I sometimes eat at times that probably shouldn't be eating, although I do try. I think the eating, it's the one that I kind of think the most about. I try to eat earlier in the evening. So I mean, I think that I do think about it. It doesn't mean that I am any better than everyone else.
Sure,
I tried.
Knowledge doesn't translate to actual action most of the time. Well, that's awesome. I guess another thing that I want to ask, just because I really want to press this button, is Glorianna, tell me about a dream you recently had.
A dream I recently had. Ooh, last night I had some very weird dreams, but I can't fully remember. There was one that I did recently that I was like, what was that about? Like, I was like, that was like such a weird. I dreamed, this was a couple days ago. I dreamed that I was walking. I was in the neighborhood where I grew up with my parents, and I was walking the neighbor's dog.
Okay.
That I wasn't supposed to be walking. My sister should be walking the neighbor's dog. And then somehow another dog came. It was this black fluffy dog, and then there was this other little white fluffy dog, and I was like walking them, and then one of them stopped to use the bathroom, so I turned around to look at the dog that was using the bathroom, and the other one went up into the street, and then suddenly I turned around, and a car had come and had cut the leash and had stole the dog.
Oh my God.
And then I was freaking out because it's not my dog, it's a neighbor's dog, and I was crying and having a meltdown.
God, that sounds not like a dream, more like a nightmare.
It was such a weird dream. I don't know where it came from, but
wacky. Yeah. I mean, maybe one day you can tell us a little bit more about zebrafish dreams.
Right? Yeah, the zebrafish dreams. I don't know the answer to that.
I literally have no idea. Okay, so let's exit our dream sequence. Okay. And we're back to the real world. Okay, before we wrap up, Glorianna, I have a couple last questions for you, more specifically towards being a graduate student. Mm-hmm. What is some advice that you would give to aspiring scientists or anyone wanting to get more involved in science?
Yeah, that's an interesting question. I will say, don't be afraid. Don't be afraid to seek people. Don't be afraid to talk with your professors and, talk with people that you find interesting. Putting yourself up there, it's, I think, one of the most important things, because if you're not out there, how can you be able to get those opportunities? Mm-hmm. So I would say, don't be afraid to send that email. Don't be afraid to, apply for that internship. The worst thing that is going to happen is that they're going to say, no, you won't get it. And that's fine. Life moves on. Other opportunities will come, but maybe you will get it. Maybe you will get that opportunity that you need to be able to do research. So I would say putting yourself out there and not being afraid to be out there, I think is the number one advice that I will give.
That's great advice. You miss 100% of the shots you don't take.
Yeah, exactly.
Classic. Yep. Okay, great. Another last question that I just have for you is, in this tumultuous time of the world, what is something that is keeping you positive in work or outside of work and making you positive about science?
Yeah. Well, I think outside of work, my cats.
Your cats. What are their names?
Persephone and Possum.
Gorgeous names.
Yeah. I love them, and being able to go home and just have the cats and play with them, and they're just I don't know, it's just the best thing ever.
Yeah.
I think that even though things are hard and science is being attacked, I believe in what we do, and I know that we do it right, and I know that we all have the best intentions, and I know that none of us are doing the things lying or making things up. So I guess maybe it's naive of me, but I truly believe that we are doing the things right. So in the long term, it will be okay, because we have the data, we have the evidence.
Yes, we do.
We have what proves it. So I feel it should, at the end of the day, it should be okay. I don't know. Maybe it's naive, but I kind of hold to that thought that this is just for now, but in the long term, we are on the right side of history at the end of the day.
Wonderful. I love that. Well, Glorianna, thank you so much for joining us today at Science Positive. It was such an absolute pleasure. Anything else you want to say to the listeners, to the viewers? You can say it in Spanish too, really do that science communication.
Yes.
That's up to you.
Well, what I will say is that maybe when you're scrolling in social media late at night, maybe you should think about your circadian rhythms, and you should put the phone down and grab a book.
Book over TikTok.
Yes.
Awesome. Well, thank you so much for joining us. And that is all.
Well, thank you.
Thank you, everyone.